Cloud.
Okay.
Welcome, Maruna.
Hi there, Dr. Cox.
Yeah.
Do you have instructions on how to send funds to you and your efforts?
Uh, that's we're still up. We applied for 501(c)(3) status. The government has shut down, and so we've been kind of put on some back burner. So I don't yet have instructions on that, and so I need to. But we have had other international um, um, kind of participants pay for gloves. And I should check with my wife Laura does kind of the shipping and receiving and things like that. And lots of you have corresponded with her. Aruna Friend says she's our first recipient of a glove from India. Um, and so I don't think I've ever filled out a customs form with so many syllables in my life. I'm I'm hopeful that that most of them are in the proper category and they get to you, Aruna. Um, yeah, I'm eager to keep up with that process, and I'll ask Laura how she communicated with the folks that that were able to pay from Japan. Another set of gloves went to Japan this week as well. So
Oh, okay. Yeah,
yeah,
and I have US dollars as well, so I can pay in USD currency. I just need to know where to send the money
gotcha,
to.
gotcha. Okay, cool. I will. We now have an Espanola GloveWorks account, and one of my fellow board members, Janice, is with us. She's one of the high school teachers, so I'm really happy that she's here tonight. Um,
Great.
and welcome, Emily and Roger. I'm glad to see you guys.
Hey, Steve.
So we're embarking on kind of part two of Meet Your Brain, in some sense. Um, we know we've all got one, and uh, I still remember when I kind of got off my old my previous career as a mathematician and started learning more about the brain and what a fascinating trip it was and is now still is. But it's especially, I think, fantastic in the early days of kind of getting a deeper appreciation of the wonders that are going on inside. Um, and I mean, you can wonder about. I mean, even as art and poetry are kind of gateways to those things. But as a scientist, to really to see that the advances we made in the last 100 years, understanding basic mechanisms is just so. For me, it's really hopeful, and I hope that hope kind of transfers, through our dialogue tonight. These it's still so it's that hope comes in parallel at a time that we're also through industrialization, we're also poisoning ourselves as we go along, right? And so Parkinson's is this thing that's a result of industrial revolution, which was again kind of an unintended offspring of the scientific revolution. And so it's, is brain science going to keep pace with kind of environmental degradation of our planet? Is this amazing situation that we find ourselves in? Um, and so the environment is a much harder thing to get our heads around. Um, the brain should be easier since it's like right there. But there's this. I may have shared this one kind of pithy comment before, that if the brain was simple to understand, we'd be too simple to understand it. So there's this so it's this kind of it's a cycle. It's like back and forth. We kind of spiral in and then start asking questions and realizing we got someplace or are we fooling ourselves? Um, and so we began last time by working from the outside in. I find that intellectually an easier place because the sensory stimuli, they're all kind of they're literally at our fingertips, right? And at the tip of our tongue. Our senses are where we engage with the world. And then things get complicated as they find their way up here. And so we might finish that discussion tonight. And then the next step will be saying, once information gets here, how does the brain plan motors? How does it plan muscles? How does it plan to engage with the world? Um, And how does the glove fit in and other possible treatments for Parkinson's? How do they um, how do they help the brain engage in new pathways? Or how do they declutter the noise that's currently there with the lack of dopamine? Um, so with that preamble, I'm gonna click the share button and welcome a few more friends. Betsy, is nice to see you, and Minna and David, welcome. So I'm going to share. Um, can close that down, and and these were the notes that we were um, kind of working our way through, and largely they're meant to be picture notes, with things that are a little bit of vocabulary, or maybe even a kind of a side link to get our hands on what's happening. So last time we
Can
saw that it's only three neurons,
you go
from your
full screen?
um, yeah, in my let me go down a little bit. Yeah, that's about as big as I can go if I'm staying here in the Google Doc world.
Okay, that's
So
pretty good.
I may be able to go. I mean, I can definitely grow the pieces. Is that a little bit better, Ken?
This Ken says it is.
Okay. So so last time we counted from a tickle, or a vibration of the fingertip. Um, Things make their way to your shoulder, just around your neck, on your spine, then up to the midbrain. They cross over, um, and a long, long neuron axon up to your thalamus, the main relay station, and the thalamus then sends it to cortex. And in cortex, there is a map of your body. In fact, there are four maps of your body which will kind of reinforce, and they talk to each other independent of our conscious awareness. Um, so leg and foot is up there, trunk, forearm, hand, face, and the rest of innervated skin makes up the rest of these cortical folds and gyri. And we began looking at different ways to kind of chop up the brain to make sense of what's going on inside. And we wanted to kind of study that pathway and largely to figure out, kind of what's happening at each step. But these three these three neurons, from finger up to cortex, they're pretty reliable in the sense, that they largely are just following stimuli. We'll see that the fingertip has different receptors that are going to be, kind of receptive to different types of stimuli. But more or less, if it touches one finger, it kind of follows kind of one pattern all the way up to that finger, even that individual finger as it sits in the brain. And so it's really up here, up in cortex, where all the action happens, where the brain receives the
Read this.
stimuli.
You should read it.
Say again. Sorry if I missed something, you guys. So So that was the gist of last time. Fingertip to medulla, medulla to thalamus, medulla in the someplace in the midbrain, brainstem, medulla to thalamus, and then thalamus to cortex. And if this picture is too complicated, then I was asking you, can you simplify it in your own terms so that it makes sense to you? And then we began to take a deeper dive into what's going on at the two ends. The two ends. So what's really happening at the fingertip, as much as we can say, and then what's really happening in cortex. Between is largely just kind of a reliable relay, right? Just literally like if you think about a relay race and track and field, just kind of hand it off. Communication is reliable. These are reliable partners. They're not going to drop the baton. Um, they're going to get your message to the right place. And so we began to see in this kind of more simplified view of just we're kind of ignoring now all the relay stations. We're looking at maybe a couple of different type of receptor cells that are just below the skin in your fingertip. And we saw we've got thousands of those, like 200, per kind of square centimeter. Crazy density of receptors under the skin. These are relaying messages to thalamus. Thalamus then sends them up to cortex. I'm going to zoom out just a little bit, you guys, so we can kind of see where this chunk of cortex came from. Can you see this kind of this gyre? There are hills and valleys in the brain called gyre and sulci. So a gyrus and a sulcus. And there is a place in this one particular gyrus, which is loyal to fingers 2, 3, 4, and 5, right? So your glove is basically activating digit 2, 3, 4, and 5, your index finger through your pinky, right? And and each of those fingers has a piece of real estate in cortex, and they are separated, and they're even further separated by the type of receptors on your fingertip that are talking to this part of cortex. And so we talked about slowly adapting sensors and rapidly adapting sensors. And roughly speaking, the rapidly adapting, so those are the ones that see a difference in stimuli. So when something different happens, a slowly adapting is something that sees kind of a constant pressure on your fingertip. And constant here can be, in the time world of the brain, a second is a long time, right? So something that happens quickly innervates this part of D3. This is your middle finger, and something slowly adapting. And then within cortex, and we'll see a little bit of the complexity today. Uh, this network is still under active investigation. But in fact, and I'll kind of just prepare you for the complexity, so that we're not going to get in exactly what's happening. But this is the part of the network that TOS says that the glove is actually impacting. It's affecting connections between these cells up here in cortex. And so not only is there a map of the brain, right, as we move, along the ridge, but then also as we go below the surface in cortex, cortex has evolved over hundreds of thousands of years, kind of layer by layer. And so we currently are six-layered beings. Our cortexes, our cortices are six layers 1 through 6. Um, and just as there is kind of spatial segregation as you go along a valley, as you go below the surface, each of these layers, each of these strata also has their own level of communication with other pieces of the brain. And we have a rough map of this, so we know layer 6 is largely sending information to thalamus. Layer 4 is receiving information from thalamus. We're going to especially get into layer 5, maybe today, but maybe next time. The basal ganglia, probably many of you know, your neurologist has probably brought this up. I mean, this is the real focus of research on Parkinson's. The basal ganglia contains the nucleus or the ganglion of cells that die during Parkinson's, the cells in substantia nigra. And so it's those cells, their death and their immediate impact on their buddies in the basal ganglia that lead eventually to Parkinson's. It is kind of a deep brain structure and not very easy to study, as opposed to doing kind of experiments up here in cortex. So less is known, although still there is a Manhattan Project for sure underway on what's going on in the basal ganglia. But it's still early days, and it's not historically it wasn't really ethical to do experiments on humans in basal ganglia. But now so many folks are undergoing deep brain stimulation that while during that procedure, some folks are then becoming parts of research studies where we can learn more about the connectivity of those ganglia. A ganglia is just a small brain structure, a nucleus of cells, if you wish, a bundle of cells that are kind of loyal to a particular function or region or both. And so at some level, it's just a picture, but it really does warrant, just it warrants our attention, right? The fact that we have these rough delineations of how information is distributed in the brain. Still too rough to cure Parkinson's, but this is on our way to understanding, what type of communication breaks down, and what type of alternate pathways might we open up to bring back better function, and the glove seemingly is an explanation, is helps us understand what's going on in cortex. And so with that, I can move into this week's new stuff, which we didn't really get to. Um, but this is the beginning of seeing there's a lot going on under the fingertip, and there's a lot going on up in cortex. Um, and so I'd like to jump into this kind of longer survey paper. And again, I guess just to remind you guys, I'm borrowing these pictures for us from this beautiful, beautiful treatment, Principles of Neuroscience, by Eric Kandel and company. And to get a better view of some of the 3D structures, I really, really recommend this kind of immersive 3D experience link. One of my colleagues, Chuck, told me last week that he found the third edition used for $7.98. So at $8, for 1,600 pages, that's like 5 cents a piece, something like that. It's amazing, and we don't need the latest edition for our cursory introduction to the brain, and so just to try to give some attribution to those figures. So I now want to, again, this is quite a long survey paper, but it fills in lots of gaps for people that would like to go deeper on what we've been talking about. In particular, we've just been kind of following the pathways, the three neurons from the fingertip to the brain. But the brain also needs to know about where the person is in the world. Um, and and people aren't really good reporters. So the brain has its own sensors inside your muscles and joints. So the brain has kind of a parallel network called proprioception, where it's measuring how fast your muscles are moving. It's measuring where you are in space, and it uses that together with touch to make plans for where to go next. And those are the plans that we believe are interrupted in Parkinson's. One naive explanation is that Parkinson's patients are making too many plans, and those plans are actually interfering with each other, and that slows down movement and causes rigidity and causes loss of balance. Um, there's probably a lot of truth to that, but it's just the tip of the iceberg. Um, and so I really think this paper also warrants more study. I will largely just kind of center in on figures to help you guys maybe try to tease out what's going on in the figures, as kind of as a first pass. And so here I've got largely just kind of just comments on what's going on in figure 1 and figure 2, figures 3, 4, and 5, and how they relate to the stuff that we just got done talking about. And so it's going to extend where we already are, right? So and please push back if I'm moving too quickly or jumping over certain things. I'm definitely moving quickly. But what are we talking about? We're trying to go from fingertip to brain. We've now taken a closer look at the fingertip. There are these fast-acting receptors and slow-acting receptors. And in cortex, there are many, many layers, and there are even many, many different maps of the brain. And the cortex is working kind of very hard to integrate these body maps and help us make the next move. And so this paper on the neural basis of touch and proprioception is also maybe even just to speak to the title, right, in primate cortex. So of course I mentioned it's not ethical to probe someone's basal ganglia, and but ethics our ethics at present don't apply to non-human primates. So most of the research in this paper is work that's been done on monkeys, which have a genome and a brain remarkably similar to ours, and so we're learning lessons from experiments made on examining pieces of the monkey brain, and we also now, for better or worse, we know how to give the monkey Parkinson's. So these are difficult truths that we have to deal with in medical research. And so these are some notes on figures 1 and 2. But let me just kind of jump into that paper. The link is there for you guys if you care to follow along with me now. I'm gonna sorry, scan too quickly, but I'll go just to the top of the paper. Neural basis of touch and proprioception. Um, this, like I mentioned, this is still an active field. This is a review paper, but it's from 2019, so it's still we still have a lot to learn. Um, and so let me bring you down to figure 1, and I hope this might give you guys a better feel um, for the complexity of what's underneath your fingertip. And so try to get kind of as big as possible. Um, and so we'll do a little bit of graphical surgery. I'm not doing this to you right now, but here's your fingertip. Do you guys see it coming out? And we're now gonna slice through your fingertip, through your fingertip. Your nail bed is here on the bottom, and right here we're gonna zoom in. And maybe I zoomed in too far.
Official, we were asking about whether any local officials have requested a National Guard. The local official I was talking about was Bob Day.
Is that Linda? Can I just ask you to? There's a little bit of feedback I'm getting from one of um, our characters. Let me just. Good. All's quiet. So there may be some things you might recognize. The epidermis is really, the outer layer of your skin. And for some of us, what pokes through are kind of hair follicles or sweat glands. This is kind of on the non-hairy side of your finger for most of us, hopefully. So that's kind of called glabrous skin as opposed to hairy skin. So these are always sweat glands going to the surface. And then these black things are kind of nerve fibers running through underneath your skin, and they're receiving input from one kind of set of rapidly adapting mechanoreceptors and 2 families of slowly adapting mechanoreceptors, and then one very deep one. And this is the one that really tosses after. These are Pacinian corpuscles. So these are somewhat deeper. We're still probably within a fold of your fingertip, right? Sorry, of your fingerprint, you guys, right? So I'm not very deep, right? I'm still kind of a fraction of a millimeter below the skin. And so that's this is all the electricity happening, under your fingertip, actively sending information to the brain, because your fingers are, for a big part of our experience, this is how we engage with the world. So these things are highly innervated. So this is a little cross section of what's under the skin, and then depending upon the type of receptors, so are they rapidly active? Are they slowly activating? Another slowly activating. Sorry, I've gotten kind of more than one picture on here. So let me kind of go back to that one. Sorry, guys, just trying to get the magnification a little better for you. And so, again, it's kind of like some of the Candel figures. These really they deserve a kind of careful scrutiny of kind of panel by panel. But I just wanted you to see that we can kind of paint the hand by what type of receptors are under the skin, right? And some of these receptors have small, sharp borders. That means they're only listening to a very small piece of real estate, while other receptors, are fairly kind of generous in the amount and the amount of stuff that they're listening to or sensitive to. And I really want to distinguish between the fast acting and the slow acting ones. So what's happening here is that there is a slight change in touch or pressure, right, or vibration at the fingertip. And this cell begins to spike. These individual these individual kind of vertical lines are spikes. This is the cell waking up, responding to this change. But you guys see that once the change levels out, the cell is no longer interested, right? This cell loves novelty. It does not love, steady state. But watch what happens here, right? Now you turn off that stimulus quickly and the cell responds, right? So this is a cell that's sensitive to change, while this is a cell that's kind of encoding how long, that I stay ramped up before I kind of went back down. Okay. And so there are cells that respond to differences, and there are cells that respond to a steady state. And the brain needs that information to kind of the steady state to know where it's at, and then the fast acting to know what's changed in my environment. Okay. So this is a much more detailed picture than when we looked at before, right? We've seen now at least 4 types of receptors. They're receptive fields, and again, it's just it's hard for us to think about these receptive fields, but if we think about vision, for example, I mean, we know that we can see better, in certain light. We can see better straight on than rather through peripheral vision. We know also we've got a blind spot in the fovea where all the cells pass through the retina. And so every sensory modality has these receptive fields, things in the world, that limit their receptivity. So it shouldn't be maybe too surprising that we have, that our hands are spotted. And these spots hopefully are full of, overlapping regions where each of these cells is trying to capture what's happening at our hands. And so as this cell spikes, right, so this is a spike in this cell, which then travels from the fingertip up the arm through one of the major nerves into your spine, hits the backbone, travels then up to the thalamus, and then from thalamus is relayed up to cortex. This spike is the baton in the relay race. Okay? And again, this is a fairly complicated baton. 3 spikes close together followed by 2 is even telling the brain the slope of the stimulus. Was it very sudden, right? Or was it gradual? This makes a big difference. So that's figure 1, going from your skin to maps of your skin, coloring your skin, and then trying to distinguish between receptors that listen to change and receptors that listen to steady state. Yeah, and to get into, and I mentioned the cells that are targeted by TAS's stimulus are these Pacinian corpuscles. These are the mechanical receptors. And these are very susceptible to change, right? They're not listening, right, to steady state. So your glove is meant to deliver basically quick bursts of change to your fingertips, and then all quiet, and then quick bursts of changes to your fingertips, right? Using what we know about the receptive field of Pacinian corpuscles. Okay? That's figure 1. Figure 2 is hopefully going to, and I know there's 2 weeks between where we're studying. So I want some of them to be kind of reminiscent of things that we've been to before. Figure 2 is not one of those. So figure 2 is something totally new. Oops, sorry, and I keep on playing with my Zoom. I apologize. So figure 2. We go down to the figure caption. These are typical responses of proprioceptive afferents. So these are cells inside joints or inside muscles, right? And so they are responding to contraction, to stretch, and to extension. And all I really wanted you to get from this, and I'm not really prepared to spend too much time on this. It's a little bit too far afield. There are questions about how is the proprioceptive network affected by Parkinson's? That's still an active field. of research. Um, kind of Conroy's observation that the glove works better while you're exercising suggests to me, that the stimulus that toss glove is giving has an extra boost if it's followed by, by appropriate or maybe inappropriate muscle movements. Um, but that's still kind of just a guess right now. But this is allowing us to see that certain cells in joints and muscle, are fairly, this was kind of a fairly quiet period, maybe a spike every half a second, and then all of a sudden a burst of activity, and then kind of all quiet during this part of the experiment, and then kind of back to some kind of tonic, maybe 1 or 2 spikes a second type of business. And so this is just a reminder that your brain is receiving inputs not just from your fingertips, but also from your muscles. And it's integrating that information once it gets up to cortex. They follow parallel pathways. And figure 3, I hope it's just maybe another reminder for us of, can I really, I think as I'm viewing this in HTML for you guys, I'm not really able to remove this right-hand side, but the right-hand side is maybe helping us with a little bit of vocabulary now and then. So this is not your brain. This is a monkey brain. It looks pretty similar to us. It has 4 maps of the monkey body here in this part of cortex, and then those maps of cortex go back and forth through the main relay station. This is the thalamus, and this thalamus is then connecting, as is the relay, to the downstream neighbor in the brainstem and the medulla. And the medulla is then connected to the backbone. And around your neck, these 17,000 fibers on each side leave your neck and go down in 3 big nerve bundles to innervate your hands and your muscles. All right? So they talk to your skin, they listen to your skin, they listen to tendons and joints, and they listen to muscle. So again, those are the kind of the 3 main cells from the hand up to the backbone up to medulla, medulla up to thalamus, thalamus to cortex. Okay. So hopefully this is review. But again, I don't know anybody that groks this stuff on first viewing. So review, and maybe seeing it in a slightly different illustration, even a slightly different animal, helps us understand. There's a little bit more detail in here that we're not going to get into. Of course, the thalamus has its slices, and it's also loyal to where you are in the body. The thalamus has body maps. So it's again a level of detail that we could enter into that I'm not going to do for the moment. So I'm going to move to the next figure, which is again hopefully I've kind of prefigured it. And so we've taken a closer look at the fingertip, these 4 types of receptors, and we understand that some of them respond to change, some of them respond to so-called tonic stimulus, something that's more or less always there. And as they make their way up to cortex, the cortex has, so this is basically 4 maps of the monkey body, 4 maps of the monkey body that are adjacent in cortex. And so let's take a look, right? So the orange part of the monkey, is this maybe big enough? Can you guys, oops, sorry. And sorry to zoom in. The HTML zoomer is a little bit odd, but we're interested, of course, in the orange part of the monkey's hand, right? So in the monkey's hand, monkeys have 5 digits, right? There's the thumb and digits 1, 2, 3, 4, and 5. And so the monkey hand is in 4 adjacent slices of real estate. And for the monkey, we even know where it is, right? We know which kind of, from which kind of mountaintop and valley to follow to understand where the hand lives in the monkey. And so that means when we do experiments by touching the monkey's hand, we know where to listen to see what effects that has in cortex. So I will shrink back on that one. And so figure 3, figure 4, these 4 maps of cortex, or sorry, 4 maps of the body in cortex. So the face and the nose, the foot, the toes, the leg, it's all there, right? And again, why 4? That's not so clear. And then basically, like, all hell breaks loose. And so this is like an ultra complicated, kind of subway map, right? For a city that's been kind of made on the fly without any like real plan somehow. So, and again, I apologize for the alphabet soup. All I want you to see is that there are too many arrows for us to really grok or keep track of. So these are arrows within these parts. So I think we got thalamus down there, but thalamus is sending information to lots of parts of cortex. So C is cortex. And these are regions within cortex that are talking to one another. So this is the complicated conversation that as sensory information makes its way into the brain is deciding on next action based on the motor plans that you've already built, based upon the best current information, based upon memory, also based upon vision and upon sound, and all these things are there's this harmony, this interplay in cortex. And this has been interrupted by a lack of insufficient amount of dopamine down below in the basal ganglia. And so I just want to show you that there are lots of connections, you guys, that are happening in cortex, and we're beginning to understand um, how these regions of cortex are talking to each other. And we're beginning to see how we can interrupt this conversation or enhance this conversation. Just before I leave this complex diagram, I mean, I don't want to, I mean, I don't want to, I don't want to be defeatist, right? Saying this like too complicated to understand. It can be understood, but just kind of one piece at a time. But just to give you something to hold on to, regions 3A, 3B, 1, and 2 of the of this part of cortex, these are where your 4 body maps are living, your distinct body maps. And so there's this communication between these 4 body maps. And each of them is also loyal to one of those 4 body maps in thalamus. So a lot of your body is loyally moved around in this conversation. But what's actually happening between these maps is still a puzzle. There are currently there's currently too much discussion, and we haven't designed the right experiments to really tease out when one arrow is important while another is not in the building, let's say, of a motor program to move across the room or to lift a cup of coffee. Things we think are fairly simple tasks, which require a significant amount of planning in the brain. And so I want to talk about if you wish, you guys can just think about picking an arrow, right? And so this is a neuron in, say, region 1 of anterior parietal cortex talking to region 2. And I want to now kind of drill down into what does it mean for one cell to talk to another? Because that is Tassa's explanation for what the glove is interrupting. Tassa's thesis is that there are too many connections between these regions, that basically dopamine is kind of a governor, and when the governor is asleep, the cells decide to party, and they make too many connections. And the glove seems to be a mechanism for pruning some of those premature connections or connections that did not really need to be there. And so where I want to take you next is an understanding of what's a connection between neurons in the brain. And then how could the glove be involved in breaking some of those connections? And apparently breaking them, with and kind of again back to Ken Toronto's, response that the only downside of the glove is that you have to take fewer drugs. So it just seems that we're breaking their connections are being broken in a way that are restoring what dopamine seemed to be doing in the first place, right? Because I don't want to advertise that we're just breaking connections pell-mell, right? I mean, quote said that. I mean, a great cure for lots of diseases is just kind of decapitation, but that's a little bit too severe. So removing connections, is just kind of on the way to de-innervating the brain, but that doesn't seem to be what's happening with the glove. It's a more selective breaking of connections, largely probably due to Tass's discovery of the stimulus pattern. Okay.
I wonder if what's going on with the task glove is similar to what happens like when you learn to walk. I mean, the brain has no experience and you learn to walk, but then you don't have to think about it anymore because those pathways have been established. In the same way with the glove, if it's affecting certain parts of the brain and eradicating some of these extra channels, that's why I think Conwar talked about you don't need the glove 24 hours a day.
Mm-hmm.
You wear it and certain patterns are established and they're remembered.
That's a great observation, Ken. Yeah, yeah. I don't yeah, to test that, right, we really have to have a glove patient, under the knife to watch what if patterns are getting created or learned, right, that are acting in that fashion. Yeah, so um, yeah, I don't that's hard. So we use the word learning, right? So that's what I'm about to show you is our best understanding of how the brain learns. In fact, and this is what Campbell won the Nobel Prize for. I think it's the most amazing part of our whole story. Um, and so I'd like to spend the next, 10, 15 minutes talking about how cells communicate.
Thank you.
How cells know then that they've learned something. Like I mean, it's pretty easy for us to kind of to use human language as if cells were human because we're made up of those cells. But cells themselves are fairly limited organisms. So how do they detect that something? How do they know that something has changed that they should remember? Uh, and also, how do we know what to forget, right? And so it'd be very difficult if we didn't forget some certain things. And so learning how to grab a new pattern, as you say, learn how to walk, but also learning how to ignore certain patterns um, that that basically Parkinson's has written into the brain. Um, and so I'm going to steal the screen again, unless any other comments or observations.
No.
Okay. Um, so kind of back to our notes, right? And so this last figure 5 is the one with, like, for me, like way too many arrows. I mean, and again, I don't mean that in some trivial sense, right? I mean, those are super important arrows. Probably each of those arrows is like a PhD thesis, right? Or like, a postdoc has spent, 10 years establishing that there is this kind of reliable connection between these two regions of the cortex. It's just that as kind of people looking for a cure, what should we learn from that? And so it's, there's this evidence for rich and so challenging interactions across cortical areas. And all I can say is that those interactions, the complexity of those, I mean, that's where perception comes in, right? That's where, or as maybe Ken was saying, that's also maybe where walking becomes automatic, right? Where we don't have to consciously load in the walk pattern, although that doesn't mean it doesn't happen, right? And so the brain must load in the right motor program. And the basal ganglia seems to be the thing that sorts those motor programs. We develop so many of them, right, for our kind of characteristic movements, that the movements define even who we are as people. When Laura or David's friends said that they saw that they were back, I think what that meant was they were moving and looking like the previous person that they knew before Parkinson's. So that's, that basic, our characteristic movements are our subprograms, our motor programs, and they're literally called motor programs that are stored up there in cortex and that are sorted by basal ganglia on the way to our muscles. Um, and so it's rich. Perception is just it's a cover up, right? For like, I don't really know what's going on there, but I do have this percept of touch. and movement. And so maybe finally, in minute 46, we can get to the glove. So I've tried to give you a feel for the complexity or the richness of that network. Um, and and what's fascinating about, a lot of applied science, you know, and especially this is also true of engineers, right? I mean, so engineers don't wait until they understand everything about the structure before they begin to change and control it. And medical scientists, do not wait until we have a full map of what every cell's doing in the body to begin to kind of poke around and see what works, right? And so Tuss's glove appears to alter this cortical network, um, and he argues again, like the metaphor I mentioned before about kind of cells making too many connections, and then kind of firing in synchrony, and that synchrony affecting your inability to make voluntary movements. And that one way to desynchronize cortex is by to diminish the synaptic weights between cells. And so to diminish the impact that one cell has upon another. And so that impact, that place where cells communicate is called the synapse. And my next picture is kind of a movie with four stills, sorry, of a movie of what's happening at a synapse. And the synapse for me is the most marvelous structure in the human body. It's especially teaching teenagers, Alicia, for example, telling them that they are not hardwired, right? You can change your brain. This neuron is not connected to this neuron. They communicate across so-called synaptic cleft. It's only a nanometer or two wide. It's very, very, very narrow. But the fact that electricity gets turned into chemistry, gets turned into electricity, this chemical kind of border zone is where everything happens. It's where everything happens. So where dopamine happens, for example, as kind of a secondary neurotransmitter. So I really want to, you know, I'm probably going to ask you to spend like 2 hours with each of these figures as we leave, but hopefully I've curated they're chosen to be action packed, and they tell an amazing story. Um, and so what happens, when say this is let's pretend, you guys, right, that this is a um, this is a neuron that's receiving information from thalamus about something you've just touched, or a vibration you receive from the glove, right? So a vibration reliably arrives up in cortex, and by that it means that cell has spiked. Remember before I showed you kind of a vertical kind of cartoon of a spike? This is less of a cartoon because this cartoon has numbers. Um, a spike is really when the voltage across the membrane of the cell changes by about 70 or maybe even 100, a tenth of a volt, right? 100 millivolts, a tenth of a volt change across a cell. So when that invades that cell up in cortex, that's a signal. It's a signal for calcium to enter the cell. When calcium enters the cell, it causes these little round vesicles to dock with the membrane and release the magic, release neurotransmitter. Oh my God, this is so amazing. A spike in the cell brings calcium in through a channel. That calcium rubs up against these vesicles, gets them to dock and release their contents into the synaptic cleft. Okay? This cell has been excited and is now releasing messengers into the world. The other side of the synapse, only 2 nanometers away, is another cell in cortex that's now going to be trying to process that vibration that happened way downstream at your fingertip. So as neurotransmitter makes its way across the cleft, it binds with receptors. Those receptors then largely allow sodium to enter the cell, which makes a small, maybe a 15 millivolt change in this cell's membrane activity. So this guy got a tenth of a volt, um, stimulus. This guy now maybe got a tenth of that, right? But can you guys imagine that if this happens at 10 different synapses, that maybe this cell's going to fire? Okay. So this is just happening at one synapse. And now again, these crazy numbers in the brain. You have 80 billion of these cells. Each of these cells has 10,000 synapses.
Huh.
And so, but at any given moment, only about 40 or 40, maybe 50 to 100 of them are active. And if in a small enough window of time, enough of them are active, it will cause this next cell to spike, and information is going to move through the network. Okay. So this is 4 stills, right? This is the same synapse, just kind of watching it. This guy, the cell on the top got stimulated, calcium enters, neurotransmitter gets released, neurotransmitter gates receptors on the other side, which depolarize this cell. This communication is entirely plastic. What I'm going to show you is that if this cell does fire after this cell firing, oh man, then this cell sends a like to this synapse and a new receptor grows into the membrane. I increase his synaptic weight. While if this cell fires and this one does not, then that means this synapse is probably not really reliable, messenger, and I'm going to prune one of his receptors, or 2 or 3. I might even prune the whole synapse. So real estate's expensive in the brain. And so here's the plasticity. Here's the change in the brain. If this cell reliably fires this cell, I will increase the synaptic weight. If this cell does not reliably fire this cell, I will decrease the weight. I will prune this. And it's this pruning that Toss believes we're doing with this glove. Okay. And so those are the words that are about to follow here. This postsynaptic cell sums the postsynaptic potentials from its thousands of potentially active synapses and fires when this sum is large enough. When an active synapse participates in the firing of the downstream cell, that cell, this is a millennial word. It likes that synapse, rewards that synapse by increasing its weight, by adding more receptors. This is fundamental to how we remember. So again, all of these things were discovered largely by Eric Kandel and his group studying sea slugs. And so that's our best model of memory, right? Is kind of the ability of a cell to know that it was involved in the firing of its companion, right? I typically get that in class by eye contact, and I'm hogging the screen, so I don't get any eye contact right now, but I hope I've succeeded, right? in knowing that you guys are on your way to kind of grokking what's going on at a synapse. Okay. That basically by increasing these synaptic weights, we are creating patterns or memories. By decreasing these synaptic weights, we're learning to forget, okay? Or to ignore perhaps a stimulus that wasn't so useful.
So maybe you can sum it up as use it or lose it.
Use it or lose it. Yep, that's good, Ken. And the way it's taught to neuroscience students is cells that fire together wire together, right? To use a little bit more kind of a rhyme, right? So use it or lose it is one, but and it's exactly the right tone, right? And what's happening. But in the brain, we like to think about things wiring, although there are no wires, there are just chemicals. And there's not any real fire, but this spiking, of a sudden voltage spike that comes into the cell, we call firing. And so let me just, now that we have an idea of kind of cells that are connected, and that there is a weight, right? I mean, each of you has tens to hundreds or thousands of friends, um, and you weight their input differently, right? Especially true of kids when we talk about family and things like that, right? I mean, so people you trust, or if you wish, you know, blogs are news channels that you trust and ones that you kind of look askance at. And so we all weight the inputs we receive all the time. I think it's a common human concern. But your cells have been doing it for millions of years. And so let's take a look at this is a picture from Tass's paper, and we looked at this a few months back, probably too quickly, but I think now we're prepared to take a deeper look. And so Toss is trying to give us a picture of what he thinks is happening in the brain of someone wearing a glove. Um, and for him, his brain is going to have 1,000 cells, right? That's it's not 86 billion, but it's also 1,000 is pretty complex, right? And so he has 1,000 cells, and he begins by just randomly connecting them up, giving them synaptic weights, you know. So if you have 1,000 cells, right, then you can possibly have 1,000,000 connections, right? If you had all-to-all connection, if every cell connected to everybody else. So what I'm showing you are kind of presynaptic cells, 1,000 of them, and postsynaptic cells, 1,000 of them in this array. And then there's going to be a black dot here, friends, if presynaptic cell, let's say 100, is connected to postsynaptic cell 110, okay? And yes, it's possibly true. It's pretty strange in the brain, but it happens. Sometimes cells connect to themselves. So you may have friends like that yourself, right? And so there's even so-called autopsies and the like. But but all we're showing you is this. This is a really kind of black picture, right? And this is at time zero, hour zero in Tass's language. At hour zero, the darkness of a dot here reflects the synaptic weight between this presynaptic cell and that postsynaptic cell. There's so little structure here, you guys, that it's quite possible that you don't understand what's happening there. But hopefully things may become clear that after wearing the glove for an hour, do you guys know what these 4 black places are?
No.
These are your fingers.
Fingers, yeah.
Oh, okay.
So Toss is playing a scrambled pattern into your brain, right? And right now, this is a 1,000-cell brain that has 4 fingers. And so what these stimuli are doing is, you know, basically it's playing a stimulus into one finger and then randomly choosing another finger. And so right here, you guys, if you can kind of see where my cursor is, these would be the synapses, say, between your index finger and your middle finger. These would be the synapses between your index finger and your middle finger. These are the synapses between your middle finger and your index finger. And by playing Toss's stimulus, he's more or less disconnected the neurons across your 2 fingers. The synaptic weights that were quite large there have been diminished after an hour of his vibrotactile reset stimulus. And then you might ask, well, then what happens after 2 hours? So after 2 hours, it seems like there's a little bit more background activity. It's a little bit crude. It's not so easy to see, but even the fingers are beginning to kind of fade away in the brain. And then after 3 hours, you know, the fingers are fading away even further. And so this gets to the question about, you know, how much stimulus is too much stimulus for the glove.
Yeah.
You know, is there a point of diminishing return? Am I just going to shut down all the weights? Yeah, which would not be a good thing. Or is this the off state Parkinson's brain? You know, and this is the on state Parkinson's brain, and the glove can bring you from off to on without drugs. And again, I'm just trying to speculate, trying to help you see the patterns that are here. Okay, that Tass is arguing, and again, this is so this is not a cartoon, right? He literally ran a model of 1,000 pretty good neurons with synaptic plasticity, neurons that could tell each other whether they were firing each other or not, and whether they should increase or decrease their weights depending upon this certain window of time. So this is a really, really well-studied model of plasticity that Toss is using. He's playing his random stimuli into that model and seeing the fingers kind of appear while he disconnects them in the cortex. And again, I know that this is not really, he's not peeling apart your fingers, of course, right? He's lowering the weights that would connect these two fingers together in cortex. He's up in cortex, breaking connections, maybe too violent of a word, but he's lowering the weights between neurons. He believes he's desynchronizing cortex by lowering these weights. And so for me, that's the conclusion of kind of our first pass from fingertip to cortex. There's lots of complexity down in fingertips, um, but it's completely, I shouldn't say completely, it's really, really well understood. We understand the whole vocabulary of receptors. We know their personalities. We know what stimuli they're receptive to. We know how they make their way to the brain, but then once they get to cortex, it's super, super complex, right? As they share information across fingers, across brain maps, on their way to giving this motion. What's key and what's kind of vulnerable there, right, um, is that we can change the network by putting dopamine back in the network. That's what you all are doing. And Toss is arguing we can also change the network by playing stimuli that somehow don't follow through, right? So these are stimuli the brain is receiving, but it's not making really fruitful connections. And since it's not making fruitful connections, it breaks connections in the brain.
Along those lines, perhaps as you talk about breaking the connections between some of the other fingers, the way everything's interconnected in the brain, that affects other parts of the body that are talking to those fingers.
Yeah, yeah, yeah.
I mean, or
That's
parts
right.
of the brain that are talking to the fingers, not talking to the feet and
Mhm.
whatever. So as I diminish the ones where the fingers are that are bad, I'm correlating that same to decrease to other parts of the brain that aren't getting the signal anymore.
Yeah, yeah. That's it's it's a jump to kind of grow Toss's model, right, from just those four fingers and 1,000 cells to understanding, kind of the bigger kind of gait and balance issues for other parts. Um, so we either need like a bigger model that allows us to think about other brain regions, or we need to make more, better observations about what's happening to other parts of your body as you're wearing the glove. It also, though, there's another. So he offers, so that what I've just showed you is a pretty fine scale, right? So we're just kind of like 1,000 neurons sitting there in a dish or on his computer, that all of a sudden then we see the fingers there, right? Because of the stimuli that he's delivering to these four different places in the network. So that's a very kind of fine scale, but still abstract because it's mathematical, a computational model. But he also offers kind of the neurological data, right? So he put his patients under electroencephalogram, and we see the intensity of these synchronous rhythms decreasing over time, as people wear the glove. And Ken, this speaks to your observation, right? So that when he does the EEG stuff, right, that brain image, he's seeing your whole body, right? You're seeing the whole motor cortex, and so it's not just. In fact, EEG can't really even see individual fingers. It only sees kind of big pictures of brain rhythms. And so these are just meant to be kind of two converging streams of evidence for a toss, of maybe how the glove is working. Um, it's still a deep mystery, right? I mean, he's he's not published much more on this. Just says he's not done a whole lot more with glove manufacturing. He has published more on these plasticity mechanisms. he has a long review paper with a number of Iranian scientists. It's a very curious paper that we could, you know, move to eventually. But I hope I brought us back to some of Tassa's earlier. So we looked at his paper early on, but again, just very, very at a high level. And I think I've just tried to help you guys get from the fingertips to the brain and to see that the plasticity of the brain, you know, is this amazing thing for teachers and for parents, of course, but then also for people trying to target a therapy. I still think it's kind of a genius of Toss to so if and I we saw those fingers appear, right? But if I played for you a billion other stimuli, we would just see junk. We would not see kind of coordinated diminishing of weights. So there's something really special happening there. And I think some of you have felt it yourself, even physically, Frank, for example. You know, but it's there. The but for me is that the effect on different brains at different times is quite different. And so I'm still after how do we tweak those stimuli parameters so that everyone has acute effects, right? Everyone um, is helped, and not just initially, right, where it wears off, but actually we can continue to tune the parameters based upon symptoms.
Steve, I put a link in the chat. There's this program I use with my students. It's called 2-Minute Neuroscience, which has some great little animations. So the one I posted, I posted one on the neuron, and then there's one on synaptic transmission, and they have another 2-minute animation on Parkinson's. It's quite basic, I mean, but in terms of the synaptic transmission one, I use a lot when I'm teaching pharmacology, and it's actually the drugs that we do have for Parkinson's on that picture of the neuron. If anyone ever wants to kind of look at it, I can kind of tell you a little bit at where the different mechanisms of action are in some of the medications that we have. Not tonight, but another time, because I do a lecture on Parkinson's meds. But I'm so excited to be able to bring to my students now that there are, you know, information about the glove as well. But in a pharmacology class, of course, we're talking about the medications,
Drugs.
but then also the wearing off at a certain point, as well as the on-off effects. So
Thanks, Margaret.
yeah.
Yeah. Cool. Yeah, I see quite a few things happening in the chat. Thank you again, Margaret. And um, yeah, Ken H question about how many receptors are being activated, you know, under the fingertip of the glove, right? And so I think there was, I think I recorded one of those factoids, right? There's so many facts to grok, but let me try to.
I think there's
scroll.
240 per square centimeter.
That's it. Yeah, 240 per square centimeter. And I think that motor, Ken, is pretty close to about a square centimeter.
Okay.
I know it's round, you know, but it's so I think that probably 240 is probably a decent, um, you know, and so it's yeah. So that's sizable, right? 240 receptors being activated, right? And so not all of them are the Pacinian corpuscles, right? Some of them are slowly active, but then and also there's this whole other proprioceptive thing, right? And so maybe as you receive vibrations though, and if you're not flexing, you know, then there's not this kind of corresponding proprioceptive, you know, kind of coincident input. So how those things interact at cortex is beginning to be teased out as well. Um, so, so I think this is a great place to stop for a number of reasons, right? So it's after 10 o'clock on the East Coast, Ken Toronto and friends. Still early in California for Pam and Frank, but it's this is the natural kind of at the beginning of the story of a more careful story, right? Of finger to brain. And then and our next set of notes, and you can please read ahead in those notes, some other action-packed figures, right, on how we get from the brain to motion. And here the basal ganglia are the star of the show. And so we really are going to have to go in. There's a beautiful 3D map that I still continue to link to. So if you want to try to get your head around where the basal ganglia sit, I just encourage you to spend a little bit of time in that 3D world. And then and magically, or maybe not magically, that the map of your body that's up there in cortex also moves down onto through your basal ganglia. So your face and your fingers continue to be mapped kind of back and forth in this conversation about which motor pattern to choose to respond to your will and to the situation that you're in. And so that should take us another full day or two. And then we'll have much more vocabulary and a much deeper understanding of kind of neuroanatomy and neurophysiology, right? So the anatomy is where are things. The physiology says how are they connected, and the beauty of the brain is they are not connected, right? I mean, they're connected across a chemical synapse, which we can change, either by behavior or by drugs or by something a little bit artificial like the glove. Okay.
Right.
Okay.
Thank you, Steve.
Thanks for joining me, friends. I'll stay on if people would like to have any kind of questions or concerns or feedback, or want to set up a personal Zoom meeting if something's not working right. Otherwise, you guys are free to go. I'll turn off the recorder as well. Good night, you guys.
Thank you.
Thank you, Steve.
Thank you.